Most of my career I've been working with, or most of my career here, I've been working with fish viruses. And one of the most important viruses in Idaho, fish viruses, is infectious hematopoietic necrosis virus. This kills millions of fish every year in the hatcheries of Idaho. And the only thing that saves the fish industry is the fact that it usually kills them before they're 2 inches long. So their solution is to fertilize 10 times as many eggs as they're going to need, and they raise up the ones that survive. It would be really nice if there was a vaccine. And that's one of the projects that I've worked on over the years, to try to see if we could come up with a vaccine that would be effective. This first slide is infectious IHNV virus, and it's a bullet-shaped virus, and it's got an outer surface that has proteins, the spike proteins, a lot like the coronavirus, except they're the spike proteins of the IHN virus. And here's a schematic of the virus that shows the spike proteins. They're referred to as G proteins because they're glycoproteins, not because they're signaling proteins that everybody else thinks about. But they're embedded in the membrane of the virus, and there's an internal matrix, and the genome is inside. One of the things we've done is to model that surface glycoprotein using software that allows us to start with a template molecule and an amino acid sequence and construct a 3D model. And the known structure, the only one that's been modeled, is vesicular stomatitis virus. But we were able to build a model using the vesicular stomatitis structure, and this is what it looks like. It attaches to the membrane by a structure here. The virus membrane would be down here. The environment, this is where the spike is sticking out. Anyway, when the virus absorbs to a host cell, it binds to sialic acid residues on the surface of the cell. And this is the cleft that the sialic acid residue binds to. Anyway, this is a close-up of a sialic acid residue, again modeled, binding to that cleft. This is a dissection of that structure with respect to the epitopes, the sites that antibodies are known to bind to the protein. There's a helix here, a sort of half turn here, and a stretch through here that are known to be binding sites for antibodies to the virus. And again, here's the sialic acid. And this is in comparison to the vesicular stomatitis protein, which is strikingly similar. And the epitopes, again, that have been reported, and again, they haven't done a whole lot with this, are on this helix and this stretch, part of this stretch through here. And we're going to see this again and again. And compare this to IHN, this stretch here, and the sialic acid would be binding in this region. Rabies is also a bullet-shaped virus and also has epitopes that are in similar areas on the surface protein. Again, the rabies has got a stretch here that is important for rabies. If we look at vesicular stomatitis from the top, we can see this stretch in here. And again, we're going to be talking about this more later. IHNV, there's part of it. Rabies, again, shows up and we're kind of running through this really quickly. Okay. If you combine the IHN peptide with, or IHN structure with, if you look at the IHN structure that's in this region that would be close to where the sialic acid would bind, the part that corresponds to the VSV epitope and the part that corresponds to the rabies epitope, there's a stretch of amino acids from about here to here that very well might interact with the sialic acid binding, or the sialic acid binding. And the hope was that if a peptide antigen was developed that would cause antibodies to be formed to this stretch of amino acids, if an antibody bound to this region, it might prevent sialic acid binding. Now, we added on a little bit on either end that are hydrophilic, so that this region hopefully wouldn't just totally form a hydrophobic blob and not be usable. Okay, so long story short, again, this is looking at it from the top, and this is the proposal that we gave to Clear Springs Trout Farms, justifying the production of a synthetic peptide with this sequence that we hoped would provide a peptide antigen that would serve as a way to immunize fish. And this is the amino acid of that region that we showed before, this region here, the amino acid sequence. Okay, long story short, and a super cautionary tale for the people who are working on vaccines right now. The fish that were vaccinated got sicker than the ones that were not vaccinated. So there's precedent for this out in the vaccine world. One of the early measles vaccine, actually, when people actually encountered real measles, they died. I'm hoping that the coronavirus vaccine attempts are more successful than this. But anyway, we had a lot of fun exploring this system. We really thought we were on the way, but it'd be nice if we knew a little more biology, we would know what was wrong with this whole attempt. But anyway, so yeah, we learned an awful lot about about modeling and about the immunology of the situation, but we're definitely not there yet. Thank you. By the way, Karen Avilia was involved with some of the early modeling. A guy named Scott LaPatra was our contact at Clear Springs Foods. Unfortunately, he had a major stroke and has had to leave science, and so that kind of slowed the project down a little bit too. Anyway, thanks for your attention.